It's a real pleasure to be here today. I've been thinking about what I would say to the group, and I guess my bottom line message is that, as you can see, connected by sugars, united by science, why we're stronger together. And I think that hopefully by the time I'm done, you'll see all of the connections and realize that if we pull together, we have a much better chance of benefiting everyone. Not everyone will be benefited every time from every discovery, but they all go into this big salt shaker called glycobiology. So let me move on and say I think the most one of the most important things, and certainly you all know this, is that if your child has been diagnosed with CDG, you may have heard that from your doctor. You may have seen it on your patient portal. And unless you are lucky enough to be one of the people who's being seen by Dr. Morava, Dr. Edmondson, or a physician who's had a lot of experience in CDG, you go in and talk about CDG to your physician, and they go, "What? What's that?" They don't like to admit that, but oftentimes that's the case. And why is that? Well, there has always been a collaboration between physicians and scientists. There's probably not enough. There's not enough communication. There's not enough times when we sit down and talk to each other. And part of that is because we use different language. Scientists will use certain terms that a physician might not typically use, and vice versa. So you may hear an abbreviation from your doc and you don't know what it means, and neither does a scientist. So you have to have that explained. So those interactions are really critical. But of course, from your standpoint as a parent, the most important thing here is the families. If we don't integrate the families into this big ecosystem, we don't go anywhere. And it's so important that all of us as stakeholders, are there and contributing to each other and communicating with each other. Well, obviously, physicians are corresponding and talking to the families about their children, and that's the primary relationship. But when CDGs first started to emerge, there were not physicians and certainly not many scientists who knew anything about glycosylation. Why is that? Well, glycobiology is seldom taught in medical schools or in even in basic science departments. So that whole piece of biology has been kind of left undisturbed, you might say, unexplored, except by a few practitioners. Well, if the families are coming to us as basic scientists, well, then we need to give something back to the families. And so the basic scientists, including myself, have collaborated very much with the physicians. And now we have as much communication as we can possibly do, but we need to increase it. And now we're all part of this ecosystem. So what's all this stuff about glycosylation? Realize that the entire surface of cells is covered with sugars. And you can almost think of it as the kelp here on the left side of the screen. And it is a big wavy network of molecules. Every cell has that. And there's some interesting facts of glycobiology, and that's all organisms on Earth have sugar chains, and they link those to proteins. Cellulose, chitin, that's glucose and N-acetylglucosamine polymers, are the most abundant biological molecules on Earth. You can see the Amazon rainforest there, and that's a good chunk of the cellulose that's produced. Of course, in your backyard, if you got a tree, that's also going to do it. So all the cells are having this on their surface, and you say, well, that's fine, but what do they do? Well, the problem, I think, has been that glycobiology was not a well-known science. And but first, I want to get into what is a CDG, what is glycosylation. I think it's important to make some definitions. So glycosylation is a cellular process. In other words, it requires intact cells to do this, and it links sugars to each other or to different molecules. And this includes all of the origins and the addition and the modification of sugars, either prior to their addition or after, because some are put on and then they're remodeled after that. So that is a very broad definition, as you might be able to think with such a broad definition could cover a lot of different molecules and a lot of different systems. And CDGs are defined as pathological conditions that are caused by genetic variants in any of the pathways that alter normal glycosylation in human subjects, in other words, in your child, or that we can show from that mutation also disturbs the glycosylation of an accepted model system, such as a fruit fly, a fish, a worm, yeast, even sometimes. So, as I said before, this is not typical that you have very many places that do glycobiology. In fact, uh, it has often been referred to as the dark matter of the universe. Well, if you're going to have dark matter, you better go to somebody who knows the dark arts. And this would be an example of one of the places that you could go to learn glycobiology. And you might have to travel quite a distance to get there because there are not all that many of them. And then, of course, here are some of our star students who graduated from the glycobiology program around the world. And you can see they are fully enamored of glycosylation and glycobiology. And of course, they're taught by uh, oldish professors sometimes who have been around the system for a long time, uh, perhaps not as old as you're seeing there, but they use unusual kinds of instruments to be able to detect glycosylation, what's going on, and what changes, and how you might be a patient within this. And they may even have little colored charts like this that they look at. So there's all sorts of different experiments, but realize that there are multitudes of sugars. Primarily, 9 of them are used for doing most of the glycosylation that we see in humans and animals, and that those 9 sugars can be arranged in hundreds, if not thousands, of different ways. So each arrangement can be very important, or in some cases, it would be trivial. And part of the challenge of this is to figure out what counts, what doesn't count. What makes a difference? What doesn't make a difference? And as we began to look at CDGs as they were coming in, uh, around 1980 was probably the first one. It was a thing called eye cell disease. And this was the first really recognized disorder of glycosylation where you could pin it down to a specific function. And that function was to put mannose 6 phosphate on lysosomal enzymes. In other words, that were going to go to the lysosome and be used for degradation of many of the macromolecules that contain sugars. Well, when that mannose 6 phosphate was not put on to those, and you can see some evidence of this in a movie with Harrison Ford called Extraordinary Measures that talks about mannose 6 phosphate. And because you wouldn't do this, all of these enzymes, instead of staying in the lysosome where they could degrade molecules, they were secreted outside the cell. And that led to an accumulation of massive amounts within the cells that were not properly glycosylated. So this was a big turning point. And by, about the end of the first decade of the 2000s, we had almost 50 different disorders. And then if you look at 2013, I mean, it was amazing. There was a new glycosylation disorder discovered every 17 days. You think about that. A new paper would come out. We became aware of new disorders. Those disorders have been around for hundreds, if not thousands of years, and parents have wondered through those decades, through those millennia, What's wrong with my child? What are these problems coming from? Well, now we begin to get an idea of what that looked like and where it could be sort of started out. Now, as the human genome was sequenced and as we got to know DNA and proteins, but we still didn't know a lot about sugars, those continued to increase up over 100 by 2013. Go to 2017, we've got 125. Is there no end to this thing? Now it's 145 by the time you get to 2020. And then now, if you look at this, it's 190 different disorders. You say, well, why all the different colors? Well, those result from pathways of glycosylation. Those pathways may be shared among a large number of molecules or a very limited number of molecules. And each one requires some analysis to figure out what that does, what that gene, what that enzyme does normally. And then how does that affect your children? And what can we do about it? Now, the fact that glycosylation occurs in all cell types, there's certainly going to be more CDGs that will be discovered. And you may even wonder, is there a limit to the number of types? Well, theoretically, there would be another 100 types that you could imagine exist out there. So what does that look like phenotypically? And you may say, well, I know my child has a glycosylation disorder, but it doesn't look like anything like my friends over here who also have a glycosylation disorder. How do we pull those things together? Well, I think you have to realize that because sugar chains are present in all cells, and they have multiple functions, not only on the surface, but in directing proteins within the cell, to stabilizing proteins within the cell, and even after they're outside the cell. And if they're not stabilized properly, they get degraded. So you go through all this work to make these things, but they're not properly glycosylated, and they begin to fall apart. When that happens, then you have a disorder. What does that look like? Well, in trying to arrange this, we put together a paper about a year ago, and we looked at the different kinds of disorders that are plotted on the left. Now, this is from a sort of a biochemical uh, perspective. In other words, what does each individual gene do for that glycosylation? And you can see there are lots of different disorders. Along the bottom, we plot what organ is most affected. And the ones that are most affected are in purple and blue. Those that are least affected are in red. Well, as you can see, there's a lot of activity in the neurological disorders. The majority have neurological uh, problems, and they can be all over the place. The other thing that's common is dysmorphic. So that means the skeleton or the face or the proportion of the limbs is not quite right. And so you can see that a lot of those are affected in CDGs. The skeleton is oftentimes um, abnormal in some ways or other. But that gives us clues as to where we can look for what these genes are doing and how they're behaving. You get over to the far right, and there are not many that have dental problems. There's some, but not a lot. Same true of hair. And surprisingly, renal disorders are fairly uncommon. It's not means that it's absent. So how do we sort out all of these different types and all of these different colored blocks representing different sort of pathways? Well, if we try naming the disorders, and hopefully that can lead to treatments, we were doing it originally by Roman numerals. Now, I don't know about you. It takes me a while to count in Roman numerals. And if you're trying to count up 190 and express that as Roman numerals, you're going to have a hard time. So the way it was traditionally done is that if there was a physician, and usually it was a physician, but sometimes it was a scientist who discovered what a disease was, then that disease would be named after them. So there's Jochen syndrome, Sly syndrome, Tay-Sachs disease, Sandhoff disease. Each one of those was caused by a deficiency of a different aspect of glycosylation. Not true for Angelman syndrome, and certainly not true of Lou Gehrig's disease. And it's not that Lou Gehrig discovered this, but he was one who first had this disease called ALS. And because he was such a famous baseball player and he was afflicted with this disorder, it got his name. And so, how did we put those together? Well, all of those different disorders, and when they were categorized, they had very shared specific general phenotypes. In other words, appearances, whether it be neurological, a combination of neurological things, or sometimes very specific symptoms. And we were able to define those mostly by organ systems. So what you would see is that there would be some disorders that would be primarily liver dominated, or other ones that could be GI. In other words, problems with growth, nutrient absorption. And as I said, the most common one would be a neurological deficiency. So there would be a delayed development. There would be um, intellectual disability. My sister's one of those, but she doesn't have CDG. Turned out I'm the carrier of heterozygous carrier of the most common mutation in CDG and PMM2. So if you're PMM2, we likely have some genetic background in common. And that makes me feel very connected. My sister, we tested her for CDG and she didn't have it, but she sure had a lot of the symptoms. Has a lot of the symptoms. She's still around and delighting my days by um, days when she's doing well and she's happy. Well, for a while, then we decided that we would use a biological biomarker test to name CDGs, and that biomarker was transferrin. And what is a biomarker? Well, it says something that's associated with the disease, and maybe you can see it being more severe in severer types of the disorders and less severe in milder types. Well, that fell into two categories, which were type 1s and type 2s. And then to distinguish those, we started using lowercase letters. So you could have type 1A, type 1B, type 1C, type 2A, type 2B, type 2C. Well, you can see where we are now with 190 disorders. The alphabet shall not hold us. We're not going to be able to get by just doing that. So there needs to be another way that we can put together some nomenclature that will be useful and that people don't have to go back and say, well, now what is Sly syndrome? What is Jochen disease? We don't know that. And so the traditional types actually now are named by giving the gene name dash CDG. And you say, well, do you know that in each case there is a link to a glycosylation abnormality that gives the results and causes the pathology in that gene? And I can say, no, we don't. But because of the definition of having to find a biomarker, a model system that mimics the patient, or having a glycosylation deficiency in the patient, we know that that can serve as a marker. So if we come along with therapies, that can enable us then to have a rational idea of how to set these things up, how to get them together, and maybe think about how we can inform therapy by knowing the gene's function. And oftentimes that will come from a scientific understanding of what that looks like. But that doesn't mean you only look at what's going on there. You should also be open to an unbiased approach. So if you have some way of determining an important feature of the glycosylation in cells or in some sort of a system, you could take all 600,000 molecules that we know of and have in chemical libraries and test those. You can also go in and test those approved drugs, which have been able to give some hope for being able to see some improvements on therapy. Now, depends on what your marker is, depends on what your scoring is. If it's growth rate of yeast, well, that's kind of a minimum. If it is a behavior in a zebrafish or a behavior or an ability in a small worm or a fruit fly, that gets you a little closer. You have to be sure that the genes that you're studying in those organisms are highly conserved from humans back to those organisms, because if they're not, you could get misled. So this can actually help scientists reveal unexpected insights. And one of the things that's been pretty successful on a relative basis is thinking back to sugars. As I said, all of this is about sugars. And so here glycoproteins, and we represent the sugars by these little tinker toys over here. These, I guess you could call them aggregates of Lucky Charms. And we know that that can come from the diet sugars you can take in all the time. Now, the primary sugar you take in is glucose. But fortunately, glucose and many other sugars can be converted into all the other sugars. There is a pathway that unites all of these sugars. So you can give this sugar, and to varying degrees, it can be converted into another sugar. So you can make sugars that you don't get in the diet, you can make them from one of these sugars and transformed into that. Also, because sugars, once they're incorporated into macromolecules, big molecules like proteins, lipids, they are turned over, they're degraded, and you can use the sugars that come out of there for the synthesis of new sugars, of new proteins. And now, when you do that, those are activated into molecules that have a little extra nucleotide on them that activates them, makes them possible to now be donors in the construction of new sugar chains. It was thought for a long time that all these guys just mixed together in one big sort of happy pool. Think of the jumping in a swimming pool or scooping out something you need. You just use what you need, and then that's it. But now we're starting to find out that those sugars are really in separate pools depending upon their source. And the size of the pools and their importance in each individual cell differs. So there are lots of different sugars, lots of different pathways, lots of interconversions. Well, that will eventually make glycoproteins or glycolipids, but there's a lot that goes into this. So what do we know? What do we know about sugar metabolism for glycoproteins? Well, it was really quite seriously studied in the 1970s, and a lot of what we know about sugar metabolism and how it gets into glycoproteins, was developed in the 1970s, and they used the best methods available. But there's limitation on those. And so the ideas that we've come forward that we even have now were oftentimes based in what we knew in the 70s. And we've made some improvements since then. Now, early on, we were lucky enough in about the end of the 90s to have a patient who had a glycosylation abnormality. And we had done some experiments with a sugar called mannose. And we saw that mannose could improve glycosylation of cells that were taken from an individual. So we thought, wouldn't it be great if you could just feed mannose to a patient and have them get better? Well, we didn't know the causes of all these things. It was back in the days when it was just one disorder, two disorders. So we didn't know what was going on. And we did some experiments in our lab where we lined up everybody and had them drink this sugar called mannose. They didn't get sick, and they did okay. They weren't patients, so we didn't know it would work in patients, but we thought, wouldn't that be great if it would? Well, we got a call a week later from a doc in Germany who said, I have a patient, and we can see that his biomarker, which is transferrin, is abnormal. Do you have any idea about how much mannose to give that might help this patient? Well, we had just done those studies in the lab. I told him give this much, this often, and the patient remarkably improved. We were all shocked. His symptoms got better in really sometimes just a week. Later, it came along. We tested another sugar for another disorder, and that was fucose. And the kid showed significant improvement in a couple of days. This was totally amazing. Since then, PGM1, other disorders have used galactose to add to the sugars. And you say, well, why didn't you just do this before? There wasn't enough knowledge of the basic science to tell us where to go and how to do it. So we have come to rely very much on what we discovered in the patients to tell us we had incomplete information about what we thought was settled biochemistry. Well, it's not. It's not settled. We're always making new discoveries. And those new discoveries point the way because we're all united by sugars. We're all integrating the science together. And because so much of the science of glycosylation still remains to be discovered, remember the practitioners, our stars that we saw from Hogwarts, uh, you could probably take all the glycobiologists in the world and put them into a small gymnasium in Indiana, at sectional time when everybody shows up because their team is playing. And that would be the world's collection of glycobiologists. So it's not like we're neurologists, not like we're taught in every place. So we need to have more participation. And that participation comes from putting together the family groups, the scientists, the physicians, and everybody talking to everybody. So we know they can have multiple effects. And sometimes sugars are now being used in combination with other drugs to improve some of the symptoms of those more common disorders. So all of the discoveries we make in CDG and looking for therapies, that may have influence on many other CDGs. And the improvements in one pathway are certainly going to have implications for the others. So AI is going to help us target specific CDG types and maybe individual mutations within those. We're not there yet, but that's what we're working on. Everybody thinks the system works like this. You start out with basic laboratory research, and then that's translated into clinical research and then improve public health. But as Larry Tabak, who used to be the director of NIH, told us a few years back, it doesn't work that way. It's like this. Laboratory research depends on what we see from patient-oriented clinical trials. We get information from population-based clinical trials. And certainly the clinical trials here, especially for CDG, have informed us and telling us we need to get to work in the laboratory. So communicating, talking about all this, it actually works and it will help CDG. Again, I think if you look at this, we have a long way to go, but we're going to get there. So, sort of an overall summary, the CDG ecosystem works best and benefits most when all of us, all the stakeholders contribute to in their own expertise. Parents, you know your kids better than any physician or any basic scientist. The primary motivations for being involved are different for different stakeholders. Scientists, for instance, are probably motivated mostly by their curiosity. But I do know that whenever we talk to basic scientists, basic glycobiologists, about the marvels that have occurred in patients, they are recruited, they're touched, their hearts are touched, and they want to become involved. And they're intellectual silos, not necessarily constructed by physicians, but sometimes it happens that way, right? One specialist may not talk to another specialist, but glycosylation doesn't care about those boundaries because everybody is glycosylating. And also, to stay in business, we have to get grants. And, it's a very, very troubling time right now for American science. And future is yet to be mapped out, but we got to try to stay in business. And what helps us stay in business is communicating with physicians, communicating with families. Where will those new CDG types come from? Well, because glucose can be converted into all the other sugars, we're even finding now that things that are centered on glycogen, that storage polysaccharide that you use and you beef up for running marathons, you want to load up with glycogen. There are many disorders involving glycogen metabolism, but that funnels in to the other sugars. And we have some insights now that we're actually seeing some benefit in some of those disorders using sugars that we thought were only going on for CDG. And those lysosomal storage disorders, well, if you're not degrading things and not breaking them down, maybe you've got an opportunity to build up and deplete some of those pathways that may rely much more so on recycled sugars. We just don't know. So we all work to do things better, and that's all going to happen by working together. And so my kitty, Siri says that, and there's that flower of hope.